Ok, te rog frumos.
Right. Deci, prima întrebare este dacă vorbesc în românește sau în limba engleză, pentru că am observat câteva nume care nu sunt de limba română.
Avem și audiență nevorbitoare de română, deci...
Okay, so in that case, maybe I should speak English.
Yep.
All right, thank you very much for this invitation, and it's always a great pleasure to speak to my Romanian fellows, and of course, I didn't forget my origins, and I'm always more than happy to be engaged in this kind of activities. First of all, can you hear me well? The volume is fine, or should I get closer to the microphone? Okay.
Yes, it's perfect.
Sorry?
It's perfect. It's very well.
Oh, perfect. Okay, good. All right, good. So, like Tudor briefly mentioned, I grew up in Romania and I graduated the so-called Facultatea de Calculatoare in Iași, and then I moved on. After graduation, I worked for 3 years at the it was called Institute for Computer Science in Iași, part of Romanian Academy. And after that, I went to do my PhD at Duke University and then postdoctoral studies at MIT. And then I came to Houston when I started my first faculty position. And last year I moved to essentially this places mentioned here. And in the next slide, I'll give you more visuals. So I'm essentially directing this new Center for Neural Systems Restoration between 3 institutions where I also hold faculty appointments. So the goal is to essentially blend systems neuroscience with neural engineering and eventually apply methodologies and particular techniques and approaches to restore the human nervous system in a disease state and a variety of dysfunctions. So during my presentation, this will be like an extended version of what I presented in Bucharest last fall. So it would probably be best if you can stop me whenever you want. So to explain certain terms or go into details if you want. I think that will help the communication between us, in my opinion. All right, so for decades, even when starting when I was a postdoc and then continuing when I was faculty, my lab was interested in understanding the relationship between information encoding across circuits in neocortex and behavioral decisions. So it is conceivable that when we essentially open our eyes, we form a sensory representation, and this pretty much is formed in the occipital lobe, talking about visual representation, but of course could be other sensory representations like olfactory, somatosensory, or motor representations, etc. And then the brain uses the representation to inform decision-making circuits to essentially make a decision, which could be move our eyes, or could be just move our head, or simply perform locomotion, and to examine the places of interest in the world. And that's how you extract information. And it is well accepted that during this process, there is both feed-forward and feedback interaction between the cortical circuits, between cortical areas, particularly between sensory areas, such as visual cortical areas, and prefrontal areas, which are also known as executive cortical areas, where neurons are correlated with our decision-making. So my lab has embarked on multiple techniques and approaches to understand the nature of this interaction between circuits, both in within local circuits within an area, as well as in a feed-forward and feedback manner, and to understand this communication between networks at the millisecond time resolution, real time, and to understand how this interaction will impact behavioral decisions. And we're doing this in my title. I have the title Complex Behavioral or Complex Behavior, and I'll get there very soon and for you to understand what I mean by that. All right, so what we realized that over the years is that understanding how electrical signals that are generated by neurons and populations propagate across circuits at different levels is not only important to understand, let's say, animal brain, like a rodent model or non-human primate model, which is our model system, by the way, work in human, in monkeys primarily. But also it is critical to understand neuromodulation and neuroprosthetics more broadly and how they're applied to the human brain. And like I said at the beginning, we embarked on a series of projects to not only perform neuroscience studies in monkeys, or even basic neuroscience in humans, but to translate some of this knowledge to understand the nature of neural dysfunction, in particular sets of diseases of the nervous system. All right, so in this presentation, I'll essentially focus on recent advances from my lab. And about 12 years ago, we embarked on essentially two sets of projects that are motivated by techniques, but on the other hand, are motivated by our desire to have a deeper understanding of the circuits underlying behavior. And the first one, and I'll start with that, is so-called microoptogenetics, essentially to dissect cortical circuits using optogenetic techniques in non-human primates. And this is very important in the sense that, if you want to understand the neural basis of behavior, now that you realize that, or we realize as a field that behavior emerges from the concerted activity of tens of millions of neurons, which are distributed across many brain areas, it's crucial to understand the circuits underlying this type of transformation of inputs into sets of outputs. And to do that, in the lab, we're using viral techniques that are essentially viruses are injected into particular brain areas, and this will facilitate us to, or facilitate the understanding of neural circuits underlying behavior. And in rodents, this technique has been very successful, but there are transgenic, say, mouse models essentially, but in non-human primates, the technique has been more difficult for a variety of reasons. So anyway, in the first part of my presentation, I'll focus on this set of projects that have to do with this direction of my lab, that has to do with microoptogenetics. And in the second part of my presentation, I will focus on, again, some of the recent advances that were made in the field of essentially more naturalistic behavior, trying to understand more naturalistic behavior, in which we are unrestraining our animals. They're free to move in their in their environment to either perform complex tasks such as foraging or simply inspecting the environment, or socially interact with other animals in order to get rewards. So for that, we need to perform essentially, or to record neural activity in a wireless manner, in which essentially we have no cables, which complicates the problems, and also to track not only the neural activity, but also the visual input and animals' behavior more broadly, and then to align all these different sets of events to understand what the animal is doing in real time at every moment. Anyway, so I'll start with the first part of my presentation that has to do with our effort to optogenetically understand or dissect cortical circuits. But before doing that, do you have any questions at this early stage in terms of like general approach or terminology, or feel free to stop me, interrupt me at any time. All right, so, okay, so in discussing my approach, or our approach, our lab's approach to understand the optogenetic dissection of cortical circuits, I will refer to some of the papers we published, particularly, I believe, two papers I'll refer to. We published in the past 2 years, right? So, but then we can go, to other sets of projects we have in the lab, but I don't think it's necessary. It's just necessary to give you, I think, a gist of what we're essentially, or just to hint on what we're doing, and then this will essentially give you the critical elements based on which you're able to form a representation essentially of what the lab is doing in this regard. Okay, so one one classical problem in, I would say, in the field of computer science, computer engineering, and computational neuroscience is the so-called stability plasticity dilemma. So the basic issue here is that in a natural environment, right, we're continuously bombarded with sets of inputs. In this case, you can see visual information, make fixations, and make tens of millions of fixations during the time course of a day, and then that's how we acquire visual information. But it's not just visual information; it's auditory, somatosensory, olfaction. So all these types of inputs are bombarding our sensory systems and transmitted to higher areas. And one classical problem in neuroscience has been the so-called, like I said, stability and plasticity dilemma, in the sense that how can we have a stable representation of the world and how the brain is maintained in a homeostasis state in the face of such a bombardment of inputs that are pretty potent, I would say. So in principle, we could imagine that, you know, neurons could, in any area, could, you know, increase firing rates. I wouldn't say indefinitely, but every input will incrementally increase responses, and then more importantly, the correlations between neurons, which are established at the time scale of tens of milliseconds, could also increase to potentially saturate the system and make it insensitive to processing new inputs when they arrive to the let's say visual system or auditory system. So how does the brain solve this problem of this this homeostasis in real time during natural behavior? And as a solution to this problem, there have been the so-called phenomenon of compensatory plasticity has been proposed, and the idea is that or the proposal has been that neural networks must balance plasticity with compensatory processes to maintain stable activity. So if there's a tendency in the system to exhibit plasticity, then there must be something that compensates or regulates that or downregulates that process so that networks are still able to maintain stable activity. And how is this possible? So just to give you an example here, this is taken from another paper, this paper published in Cell by Genetic Journal. When neurons are co-firing in a, I would say, natural or even artificial environment, there is correlated presynaptic and postsynaptic firing of cells, which in principle lead to synaptic strengthening. And this has been demonstrated in the lab, particularly in vitro, in so many labs that now it's in the textbooks. Now, during this process of so-called Hebbian plasticity, one can think or can imagine that LTP, long-term potentiation, could be induced, right? So in principle, you can have stronger responses, which, you know, in principle could increase the responses if you imagine the cascade of processes from the first synapse to the second to the executive area. And then in the end, you can have really, really strong responses and correlations that could, like I said earlier, saturate the system. And as a prevention of this disastrous consequence, you see here in panel C, synaptic scaling has been proposed to prevent sign-constrained potentiation. And you know, exactly how this is implemented is unclear, but you can think of inhibition in the brain or in cortical areas and local circuits that would regulate, but exactly will regulate this increase in excitation that could presumably increase firing rates and correlations, right? But exactly how this is accomplished in the brain has remained mysterious. So just to say one more point, make one more remark about this compensatory mechanism. In experimental studies, these compensatory processes have been described to operate at a time scale of hours to weeks, okay? A variety of studies. Yet, in modeling, in the modeling arena, modeling world, this compensatory mechanism has been assumed or shown to exist the time scale at much shorter time scales, between milliseconds to seconds, which makes sense because if you want to have an effective compensatory process in real time to prevent or to aid during natural vision or audition or anything you do in the natural world, one can think about such processes operating at much shorter time scales, right? Even milliseconds or seconds. whether and how this is possible and how is this implemented in the brain has not been clear. So we asked this question in the lab: are there such compensatory processes that operate at shorter timescales than previously believed? And we performed this study, which was led by Ariana Andre in the lab. It was recently published last year in November, I believe, came out, in which we specifically injected a lentivirus construct in primary visual cortex among human primates, and we presented essentially pulse light at a particular frequency described here between, in this example, it's 35 hertz, 10 cycles, and the animal was fixating at the central point on the screen and will receive reward after receiving the stimulation. And stimulation was paired sometimes with visual stimulus so the monkey could maintain alertness, could perform a task, etc. So our first thought and assumption was that during successive presentation of this periodic light stimulation, okay, so just to give a step back here, so what we did in this case is that we injected this viral construct to render individual neurons sensitive to light. So essentially, we were able to activate neurons in response to light. Okay. So in this case, we had a laminar probe inserted in the cortex. And in this case, laminar probe had between 16 and 24 contacts, or 32 in other experiments. You can even go higher if you want. And then we coupled the laminar electrode with a fiber optic that will emit blue light. And we were able to titrate the depth at which we illuminate the cortex, essentially, which neurons are illuminated. In this particular case, we tried to target as many neurons as possible, right? So what we did was to have this pulse stimulation for, I believe, 300 milliseconds, right, in each trial. And then we repeated this pulse light stimulation across hundreds of trials. And the hypothesis was that during this repeated stimulation, we'll be able to induce plasticity in the network so that network correlations and responses eventually will be elevated, right? And then the question that we asked is whether in real time, during the time course of a session, while we're doing this performing external stimulation, whether there is a compensatory process that unfolds in real time to downregulate the process of synaptic increase or correlation increase that is expected to be observed based on in vitro studies, for instance. Okay, and this is two examples, Unit 1 and Unit 2, in which we're representing them across trials, early and late trials, and each dot represents an action potential from one cell. Okay, so what you can see here with this like stripes, these are the elevated responses in response to light stimulation. So what was, I would say, novel here compared to other studies was that we analyzed the time course of responses and correlations, correlated activity, both during the time course of a trial and across trials, right? So in time, so we analyzed the temporal aspect of these changes in neural responses, not only in single cells, but also at the population level. Okay, so now we employed what we call so-called reverse psychophysics, and this is where I'm targeting Tudor, who said that I'm a psychophysicist. Yeah, we do psychophysics in the lab. Psychophysics is basically, it's a technique. It's a way to expose humans and animals to a particular set of stimuli and to look for a response, behavioral response. Right, but this doesn't necessarily assume that there's an investigation of neural basis of the psychophysics. But what I want to say with this reverse psychophysics is that most psychophysics studies or most neuroscience studies essentially look at the correlation between the neural response that you're either triggering or recording and the behavioral response. In this case, we did the opposite, in the sense that the stimuli were presented only with the idea that the animal will perform a task and alertness will be maintained. But in fact, we analyzed particularly the blank trials in which there was no stimulus on the screen, because only in those trials in which there's nothing on the screen, this light stimulation that is periodic is most effective in inducing plasticity, because otherwise the effect will be washed out by the effect of presentation of stimulus. And the combination of optical stimulation and stimulus presentation will essentially destroy this temporal relationship, the periodicity of the stimulus, and will render this light stimulus ineffective in inducing, I would say, strong change in the network. All right, so this was our working hypothesis that will induce this plasticity, and then presumably we'll observe this compensatory process if it exists, right? We'll be able to measure it somehow. And that's exactly what we did here. So here on the left, we see that what is represented here is the population activity. So this is the activity of single neurons average across all the cells in the sample that we had. We had more than 100 neurons across the sample, or even more than that. And so here is the firing rate in the control trials. Control trials means those trials in which there's essentially nothing presented on the screen. The animal was fixating on a central point on the screen and recorded continuously responses. In the so-called laser trials, these are the trials in which, and again, they're randomly interspersed with the rest of trials. These are the trials in which you presented the light pulses, and what you observe here is an elevated response as expected, which was sorted based on trials, early and late trials, and you see that there is no difference in the response of the neural population in terms of spikes per second to this laser stimulation, essentially. So there is an initial strong increase in responses, and then gradually there is I'd say, a slight depression of responses, which is typically observed whenever you have this continuous stimulation, could be fatigue or a small adaptation, and then the return to the baseline. This is captured also in this more bar representation, population activity, looking at the early and late trials. We need to see remarkable stability in the neural responses in essentially describing this effect that I just mentioned. Okay, so then the next question is what happens with correlated activity between neurons? We know that correlations are important. Pretty much they emerge from common input that cells receive from their neighbors, and this source of variability is essentially really not that much variability in the sense that it is established by local circuits. So local excitation and inhibition sculpt the shape of this correlated activity, and they've been linked to I'd say multiple, like a vast or complex phenomenology in brain circuits such as adaptation, learning, performance, and a variety of tasks. Like, for instance, strong correlations or high correlations are not really beneficial in the sense that if neurons are strongly correlated, then you reduce the accuracy with which stimuli can be represented in networks. So during wakefulness, you want networks to operate in a more desynchronized state, which is really a hallmark of wakefulness. This desynchronized cortical activity. And by the way, it's not found only in the cortex, it's found in other areas as well. So anyway, so going back to exactly what we did, we found that this green area represents what I just shown on the left, which is the fact that at the population level, neurons increase their responses across trials, right? So this corresponds to the light stimulation, right? And responses are elevated. But now if you focus on correlations instead of responses and represent them at the same time scale using the same metric, what we found is that there is an early increase in correlated activity between neurons, right? Which in fact would be expected based on Hebbian plasticity. But then surprisingly, during the time course of the session, we find a decrease rather than increase in correlated activity. So you find this large blue area, which corresponds to a negative change in correlations, which is captured here in panel G by this pairwise population correlated activity on the y-axis as a function of triad blocks, right? So let's say early trials and late trials. This is in blocks of 5 trials, and it's incrementally moved in the time course of the session. And basically what we are seeing here is that comparing the laser and control conditions, again, control meaning that there is no laser stimulation or optical stimulation. And laser means that we have the stimulation that is repeated across trials. What we found is that, this early increase captured by this green blob here And then as stimulation continues, so during the time course of a session, we find that gradually correlations decrease in time rather than increasing or even remaining stable, which suggests that this compensatory processes by which the initial increase in correlations is essentially downregulated by this, or through this decrease in correlations to maintain essentially networks in a desynchronized mode and ready to process new stimuli is maintained, which has been one of the hypotheses of computational work. I think computational models, they are exploring different kind of mechanisms through which this compensation could take place, and now we have revealed it in real time. All right, so summarizing all this data that I presented so far in these panels on top of the slide, okay? So what I want to say is that this compensatory plasticity that we observed is state dependent. What I mean by that is, by state, is that the brain could, or during the time course of a day, we go through different states of wakefulness, or even wakefulness itself could be considered one major brain state. And during this brain state, during the state of wakefulness in which I was performing the task, obviously is awake, we found this early increase in correlations, and then followed by this decrease in correlations, while responses of neurons are robustly increased by the stimulus, by the opportunity stimulation. Now, this compensatory plasticity breaks down or collapses in the sleeping state. So we had parallel sets of experiments in which the wake state was followed by the sleep state or rest state, in which animals were resting or sleeping for between 20 to 30 minutes. And we are trying to compare this compensatory plasticity or strength of this compensation process as a function of brain state. And we found that these effects essentially collapsed during the rest state. So essentially, if we're comparing correlations to in the rest state across trials, early trials to late trials, we find that we only have essentially this green zone, which corresponds to, look at the z-axis here. We find that this corresponds to an increase in correlations. So this is probably best represented here in panel H, in which between early and late trials, we not find significant decrease in correlated activity across the network. So all this indicates is that there's a major brain state dependent compensatory plasticity. So in other words, while during wakefulness, we need this state of compensation to maintain networks alert in the face of new stimulation, during rest, when obviously we're not processing new stimuli, you don't need this plasticity, you don't need this compensation, and as a consequence, you can maintain a state of high plasticity without any detrimental effects. And the brain is essentially comfortable to go into this mode in which compensatory plasticity doesn't, or is not effective, right? And then, this raises other questions, which is what could explain this behavior, this switch between awake and rest state, which we find very intriguing. And to do that, we employed computational modeling, and this was done in collaboration with Kreso Josic at the University of Houston. We do computational neuroscience in the lab too, but it was comforting to use the recent model, the large-scale model of excitatory inhibitory interactions. In they applied it to visual cortex, but the model could be generalized to other areas in which neurons were sparsely connected. And there was all kinds of interaction. So excitatory cells project inhibitory cells, which in turn project to excitatory neurons, and there is recurrent excitation and recurrent inhibition. And then one assumption in the model is that, so different type of conductances and synaptic strengths were specifically modeled in the model. So during rest state, in agreement with experimental data, one of the assumptions was that all synapses are reduced compared to the wake state, which is one, let's say, one hallmark of synaptic efficacy, let's say, during sleep. Synapses are downscaled. There's been plenty of lines of evidence showing that this is indeed the case. So anyway, to make a long story short, this modeling approach allowed us to examine different types of mechanisms that could potentially explain our results. So in a model, it's very easy to change excitation, inhibition, to change pretty much everything. But then what we found in the model is that what is crucial to explaining the switch between the wake and rest state, and even to explaining this compensatory process in the first place during wakefulness, is inhibition. So strong inhibition is effective in particularly inhibitory plasticity. So the plasticity of inhibitory synapses, right, is crucial in explaining this compensation process that we observe in time, during the time course of a session, but also the switch in this manifestation between the wake and resting state. And I found that this is quite interesting. So particularly it's inhibitory plasticity that is responsible for this behavior, but not excitatory plasticity. And I would like to say that, models are always useful, but in reality, nothing beats the real data, right? So now in the lab, okay, at the time when we performed this experiment, we were unable to control or to measure response of inhibitory cells. So this analysis was done with model neurons. Now we have this ability. We have this DLX promoter in the viral construct to particularly target the GABAergic neurons. So then now we're able to do so-called auto-tagging and to tag each neuron corresponding to whether they're excited or inhibitory, and then to see whether this mechanism is actually at play here. And this is exactly what happens as hypothesized by the model. So this takes me to another phenomenon that we uncovered in a paper published the previous year, 2022, by Natasha Karas. Now she's a resident in neurosurgery at Cornell in New York. So in this project, which was essentially completed a year before this study I just presented, we asked a very simple question that surprisingly no one has asked before. And I'll tell you why this is surprising. So we asked the question of how do electrical signals propagate in the brain? Okay, so can you actually measure this process? Can you measure the degree of propagation, how far they extend, right? So for instance, in this example, optogenetically, you were able to stimulate a small number of neurons marked here by blue, okay? And using a fiber optic that was coupled to a linear array. This was in primary visual cortex again. And then, so hence we had a set of stimulated units, directly stimulated units, and a set of non-stimulated units that were never stimulated, which is reflected here in the wake state. So we initially assumed that given the vertical circuits that are established between, within a column in cortex and, these are columnar recordings, because linear probe is perpendicularly advanced through the brain tissue. We expected to find this activation across the entire column. So essentially, when we activate a small number of neurons because of feed-forward connections and also feedback within the column, we expected that the unstimulated units, I was thinking non-directly stimulated units, would also become activated indirectly, so that we'll see activation of the entire column in time. But that's not what we saw. What we observed is that when you activated a small number of neurons, the pattern of activity remained local to the stimulated neurons, while the non-stimulated cells were pretty much more or less silent. This is the wake state. However, when we repeated this experiment in the resting state, animal again was resting or sleeping for 20 to 30 minutes, we saw, as you see in these examples on the left and on the right, this rest, which was essentially performed after the wake state, right? We repeated this manipulation. We found that the entire cortical column was activated. So essentially, we see this red bands or strips that essentially sweeping through the cortex, which indicates that the propagation of electrical signals is much more widespread during rest compared to wakefulness. And I keep saying that most of the effects in neuroscience are in the 20% range, 15 to 25, 20% range. This is a huge effect. It's very, very, very strong. We found it in every single session, animal, every single time we do it, even the conditions are not optimal, we see it, you cannot miss it. And we're surprised that no one else has observed it so far. And I guess one reason why we see it, other people didn't see it, is because it's, I would say, easier to titrate the light compared to electrical stimulation, which has been the, I would say, golden tool in stimulating the cortex. And the implications are pretty important, I would say profound, because neurostimulation, prosthetics, etc., anything you do with external intervention in the human brain depends on how signals propagate, how robust this process is, and the consequences that this propagation could have on the rest of the network. And this is not well understood. So the way I link this part of this project with the previous one I presented before is essentially what happens in these two different states, what happens at the level of networks, how are neurons coupled, how was excitation expressed compared to inhibition in these two different brain states. So what we found using Natasha's project is that we found two things. First one is that we see here the word population coupling. So we computed the extent to which local neurons are coupled to the rest of the population using spike triggered average represented here on the y-axis in the two states, wake and rest. And what is shown here is that there is a significant difference, essentially in the sense that neurons are more coupled to their neighbors during the rest state compared to wakefulness. So this goes in the direction of previous statements that networks act more in a desynchronized states during wakefulness, but not during rest. And we found this difference not only in the control state, but also in the laser condition in which light stimulation was presented, right? So I guess the number of spikes that are elicited in this process was higher because now you present laser, so you activate neurons as opposed to being on the resting state. But nonetheless, the degree of coupling between neurons and local population activity is maintained high during rest, significantly high. But if you look at the magnitude of this effect, this is a huge effect compared to wakefulness. And more importantly, we're able to compute the so-called EI ratio, or the firing rate difference between excited and inhibitory neurons. So in this particular analysis, we distinguished different cell types based on the spike waveforms. So narrower spike waveforms, also based on the firing rate, etc. They're identified as inhibitory neurons or putative inhibitory neurons, and the rest of the cells are putative excitatory cells. And again, now we have the technique to directly target inhibitory neurons and to know exactly which one is inhibitory, which one is excitatory. So you can do this using opto-tagging, so more precise metrics. But the main result here is that the so-called EI ratio is lower in the wake state compared to rest, right? So what it means is that, or what we found is that both excitatory and inhibitory responses are higher during wakefulness compared to rest, but the ratio is such that the network is more inhibition dominant during wakefulness and more excitation dominant during rest. And this is essentially what explains the differential effect on signal propagation, right, during these different brain states, wakefulness and rest, but also the fact that inhibition, which is, again, stronger based on this representation, stronger during wakefulness compared to rest, is essentially at play here more during wakefulness compared to rest to downregulate correlated activity in the network and to maintain this homeostatic state in which there is a compensation to compensatory processes unfolding in real time to reduce the degree of plasticity in the network. Yeah, so this is essentially the way we think about circuits, and this is kind of our take on local circuits, right? How we can use optogenetics to understand the operation of local circuits, and potentially these results could have an impact on behavior. Do you have any questions so far, or I can go on to the next part? Yes, you have a question to that.
Yeah, I have a bunch, but
a bunch, okay.
So it's not clear maybe you said I missed, where is the recording array relative to the stimulating array? Is it in the same position or?
So it's not a stimulating array.
Sorry, the fiber optic, yeah.
Fiber optic, yeah. So they are very close to each other, within 1 millimeter of each other.
Okay.
But
and what layers?
Microns respect each other. So imagine this laminar array that has a number of contacts between 16 and 32, and then coupled to it, very, very close to it, is a fiber optic that is independently moved, right? And we stop at the point. Could be, I would say, more or less an arbitrary point that is chosen based on our ability in pre-recording trials to activate as many neurons as possible, right? And when we see that happening, because you see it at a glance on the screen that when it happens, we stabilize and let whole preparation rest for, I don't know, 30 minutes to 1 hour to make sure that neurons are stable, nothing's changing in the network. And then you just perform your experiment, which lasts between 90 minutes to 2 hours.
So how are you able to distinguish stimulated versus, infected, I suppose, versus uninfected neurons in your previous experiment, where you were showing some units that were not, you had the blue units and the black units. I'm not sure whether I understood this correctly.
So the blank, okay, so there are two different issues here. When you inject a virus in the brain in an area, not all neurons are transfected. Okay?
Sure.
It's only percentage of neurons. Percentage is pretty high. We've done this study, the histology study. We published this in eLife and along with other experiments. So we found that more than 50% of neurons are transfected. Now, the way to check for that is you shine light and see if there's an evoked response in those cells.
Yeah. Okay.
If there's no evoked response, then it means that they are not transfected when light is very close to them. And we do that across the laminar electrode to see, which neurons are transfected or not. So what I mean by control conditions, so going back to maybe this slide here, control condition or control trials are those trials in which it's just a blank screen and there's no stimulation. There's no optical stimulation on the screen or visual stimulation. So neurons are recorded at their resting action potential levels. So just emitting spikes in the resting state.
So that's clear.
When I say resting, I don't mean sleep. I mean like nothing on the screen. Yeah.
So that's clear. But in the second experiment, you had these where you had the awake versus here. Stop. Yeah. So what are the non-STIM units? How did you identify those as, I presume the STIM are the ones that are infected and the non-STIM are the non-infected, or what?
No. So, no. So it's not that, because if you want to stimulate these units directly, you can stimulate them. So if the fiber optic, so blue light is not as penetrating as red light, for instance, right? Has a higher wavelength, right? And it's pretty much local.
Okay.
So we in a paper in 2019, we actually measured the extent of
spread of the light.
The light spread and to what extent or how many units at what distance can be affected by light, and we found that that's a level of hundreds of microns. So if you have a tip of a fiber optic at a particular location and light is emitted at the tip of the fiber optic, right? You're only stimulating the neurons in a sphere around this area, right? So you're not going to stimulate the neurons that are higher.
And you record those as non-STIM. And those are your non-STIM.
Right, it's non-STIM, exactly. Okay. But if you want to elevate the fiber optic to this location, you can definitely stimulate this so-called non-STIM. So this doesn't mean they are not transfectable or not transfected. But what it means is that they are not activated directly by light.
Okay. So now, did it matter in which layers you were?
Yes, we did the study across layers, and we found that there's no difference across. It's one of the figures in the paper, I believe, the last figure. We did this laminar analysis, and we don't find differences across layers. So we believe this is a general effect. It's a general phenomenon that is pretty much found everywhere. It's We expect this to be found essentially in every brain area you're going to look at, even hippocampus. It's a part of the tissue operates in this manner. Whenever we have excited inhibitory neurons that are coupled, you're going to see this.
Okay.
Yeah. Right. Other questions? All right, so just a preliminary conclusion here for this first part of the talk is that the seminar is that we revealed this state-dependent compensatory plasticity in vivo, and in vivo conditions have been notoriously difficult to examine because of high spontaneous activity of neurons, and synapses will always operate in a depressed mode. So it's very hard to even induce any form of plasticity in vivo. That's why people even stop looking. But nonetheless, we revealed this compensatory plasticity that operates at time scale of minutes, so it's not seconds, but because initially you find this increase in correlations, and then it's compensated within minutes by this decrease in plasticity. Inhibitory cells were hypothesized, were found here to play a key role in the rapid homeostatic process. And inhibition, not surprisingly, is generally found to be the essentially gatekeeper in the brain that improper level of inhibition immediately is reflected into epileptic activity, other forms of abnormal behavior in the cells in wide range of brain areas.
Deci, hai să... Păi a fost 1400, a fost numai ce să vede ce s-a montat. Da.
Is there a question? Yeah. Or... I cannot hear you.
Sorry, that was just an unmuted microphone.
Oh, okay. So let me... Let me continue here and see what. All right, and then we found robust propagation of signals during sleep, but not during wakefulness, which again I think has consequences for maybe the way we should perform neurostimulation in the future as a function of brain state. And then one question is that whether this results generalize when replacing optogenetic stimulation with the sensory or natural stimulation, and this remains to be seen.
Nu. Eu nu văd. Zic că am microfonul deschis, dar nu-l găsesc. Stai din clasă. Așa. Deci, un mic de pus.
Is there a question? Tudor, you're saying something, I cannot hear you.
Yeah, I was trying to tell. Domnul profesor Zăgrean, erați activ și puteați vorbi. Dacă aveați o întrebare, puteți să puneți întrebarea. Acum iară sunteți cu microfonul blocat. Okay, let's move on. Maybe he will figure out the...
Sure. All right, so we saw in the previous set of slides how we think about local circuits, how we can employ optogenetics to really understand, different manifestations like plasticity, excited inhibitor interactions, propagation of signals in local circuits. So the effects I presented so far were local, right? So they only affect neurons in the local network in a reduced, I would say, scale of few millimeters. Now we have the ability, where we demonstrated the ability in our previous studies, and also ongoing studies, to examine long range circuits, not only short range circuits. And I want to give an example in this slide. One of the long-standing hypotheses is that feedback projections, right? So we know that feedback is important in the brain. So first of all, feedback is ubiquitous. there's a massive feedback that, so feedback means that higher brain areas will send inputs to the lower brain areas, right? There's a massive, let's say, cortical geniculate feedback, and general feedback projections. The fibers outnumber by a factor of 10 the feed-forward projections. So they're massive, and they're even stronger than feed-forward projections. So it has been serious, the role of feedback. Why do we need feedback at all? This slide argues that feedback is important because, for instance, attentional modulation could be transmitted by feedback signals. And this is taken from a review paper by Harris and Thiele in 2011, in which they depicted circuits of arousal and attention. And the point of the slide was to say that there's overlap between them, and they're both carried by cortical feedback. So in this example, for instance, in relation to attention, and we all know what attention is, right? So in the world we're, like I said, bombarded, essentially using this word I used earlier, by different stimulation. And if you don't filter information by attending to particular locations in the visual space or auditory space or wherever to extract information, then we cannot really deal with this bombardment of stimulation. So attention allows us to restrict processing to a particular location, and we're talking about visual attention, for instance, right? In the visual field, and only extract information from one part of the visual field and ignoring the rest of the information that goes outside the so-called spotlight of attention. Now, attention is generally believed to originate in, or to be a hallmark of higher brain areas, such as, for instance, frontal A field, which is the prefrontal cortex. And we know that anatomically there are monosynaptic projections from frontal eye field to area V4, which is a mid-level visual cortical area, and then visual cortical area V4 also monosynaptically projects back to primary visual cortex, okay, to presumably carry out these attentional signals, which are essential for our well-being, right? And we know what ADHD patient, we know that is not able to effectively deal with the wealth of information in the environment and be overwhelmed easily, for instance, just to give one simple example. So understanding this circuits for attention is crucial to understanding the neural basis of attention. And this idea of cortical feedback, essentially carrying attentional signals to lower areas to make them more ready to process incoming information, okay, or selective process incoming information, has been a long-standing idea in the field for decades, for at least 30, if not 40 decades. So, however, to really understand the involvement of cortical feedback in attentional modulation, one has to perform more causal studies or investigation in which to presumably inactivate cortical feedback. And this has been notoriously difficult so far. And many scientists have employed a variety of methods, from cortical cooling to electrical stimulation to even ablation or lesion, which all leave unwanted effects, and they are not able to cleanly study this effect as it unfolds in real time. So anyway, in a relatively recent project, we attempted to answer this question, to investigate the causal role of cortical feedback and attentional modulation. And as a model system, we took the monkey visual cortex, and in this case, we know that primary visual cortex projects to a mid-level visual cortical area V4, which is another visual cortical area that processes more complex stimuli. And there are feed-forward projections from superficial layers that indirectly target middle cortical layers in area V4, and then in turn, the deep cortical layers in V4, infragranular layers, send monosynaptic feedback projections to superficial primary visual cortical areas. So we know that this anatomy exists, it's well established. Now, we reason that injecting a viral construct, in this case inhibitory construct in area V4, okay, using JAWS, I'll describe the construct later. This construct will be transported to postsynaptic terminals in area V1, okay, such that if you shine light in V1, we'll be able to inactivate specifically the feedback inputs to V1 neurons while leaving the intracortical and feedback inputs unperturbed. This was our hypothesis, right? So essentially shining light, right? This is V4. So we have a laminar electrode in V4. We have the virus injected in area V4, right? Using, again, fiber optic and red light this time. And red light is very penetrating. Red light really goes through across many layers, essentially. And then the virus will be transported, and there's evidence for this process to happen, and we demonstrated this is true in primary visual cortex. And then again, we are able to record neural activity concurrently in both primary visual cortex and area V4 to concurrently investigate neural activity during this process, okay? And the idea was that if you shine light in area V1, okay, what we'll be able to do is to, red light in this case, we'll be able to inactivate the cortical feedback inputs to primary visual cortical neurons while leaving the rest of the inputs, either intracortical or feed-forward, unperturbed. So in this case, we'll be able to selectively test causally the role of feedback inputs while not affecting the other type of inputs, which has been, I would say, impossible with using other techniques in the past. During this process, we employed this what Tudor like a psychophysics approach in which the animal was performing a task in which stimuli were presented, low contrast stimuli on contralateral sides of the screen, and the stimuli were either attended or unattended, and we alternated the two sides. And we also alternated the control and laser condition. By laser meaning inactivation of cortical feedback, and control meaning cortical feedback is essentially untouched, right? So just a blank. It's no stimulation. And first we look at the behavioral performance of the animal. Again, in this case, we had different stimuli, different contrast levels between 0% to 0%, meaning nothing on the screen, to this was about 15% C4, which is a high level of contrast that the animal will always see. And then behaviorally, we did not see any difference between the laser and control conditions. We didn't want to induce the behavioral effect because we didn't want the animal to get frustrated by not getting reward in this task, and then inducing confounding effects, right, by stimulating, by inactivating cortical feedback. All right, so this is a pretty good depiction of what was done to the animal. The color of fixation spot indicated which side will be attended, either left or right, okay, and we alternated in a random manner. And 50% of the trials had optogenetic inactivation of cortical feedback. Just showing you the results here, both in single neurons and populations. Again, this is primary visual cortex. We found that in single cells, effects are small. That is, when we shine light and inactivating feedback, at higher contrast, we found a difference in firing rates between attended and unattended conditions. So the difference was significant here, but then when light was turned on, the advantage that is conferred by feedback inputs is pretty much lost. At the population level, we're able to decode the stimulus presence or absence, and decode their accuracy in the, I would say, when we shuffle the stimuli is 50%, and we found above chance discrimination performance, both for low and high contrast stimuli. You see for high contrast stimuli, you find higher decoder performance compared to low contrast stimuli. In the control condition, control meaning that there's no intervention being made, so the network is intact. When we presented light, though, what happens is that the difference between attended and unattended sites is pretty much is gone. So essentially, the population of neurons cannot. We don't have any advantage of attention in this case, right? So because you're inactivating cortical feedback. And you show a manifestation of this effect in single cells, here showing that in the laser condition, essentially the attentional modulation, which is a percentage change between attended and unattended sites, is pretty much close to zero when lasers turn on. These effects were also found in area V4. So like I said, V1 projects to V4. And in a number of sessions, we recorded in both areas simultaneously while performing this optogenetic manipulation. And to show you here an example is that we found a significant difference in the control condition between attention and inattention. Okay, so V4 responses are elevated by attention as expected. But then when you shine light, that is when we inactivate cortical feedback, this difference is lost. So if you look here, attended laser, unattended laser, the two traces overlap. This is population average or multiple V4 cells recorded across sessions, and we find that, as summarized here, the effect due to attention is lost when a laser is turned on. The effect is much stronger than the one in area V1. You see here that both in single cells and populations, numbers are higher, and also the attentional effects here are expressed at wider range of contrast compared to V1, which is not surprising because V4 is closer to attentional, so-called attentional areas, higher level cortical areas, and the effects are expected to be stronger. But then importantly, when you shine light to inactivate cortical feedback, if you look at here, these red circles, the attended and unattended sites become, there is a small difference, but it's not significant, okay? And we can conclude from this that by inactivating feedback, we're able to abolish attentional modulation in visual cortex. So in V1, and the effects propagate to area V4. And in this paper, we have some other effects being discussed, such as the gamma synchronization, both in V1 and V4, but I'm not going to cover it here. So just to conclude this part of my presentation, we found we are interested in the lab in not only understanding circuits or local circuits, but also long-range circuits. So in this example, cortical feedback carry attentional signals, and using this method, one is able to dissect the role of the circuits, understand their role. And we only touched on this, right? We only focus on firing rates and decoder accuracy, but one can ask questions about the communication between cortical areas, right? How well V1 communicates with V4, which I didn't touch on, but we have the data, and we're currently analyzing this phenomenon to understand the communication between areas. All right, so I'm ready now to go to the second part of my talk, the one that analyzes behavior in unrestrained animals and freely moving macaque. Do we have any questions for this part of this presentation? All right. All right, so let's see. So more broadly, we know that in pretty much in every species, traditionally behavior has been investigated in animals that are restrained. When I say restrained, I don't necessarily mean head restrained, body restrained, but limited degree of mobility. Even in so-called freely moving experiments done in rodents, you have these are tethered recorders, right? So in which you have a long cable, thick cable that is attached to their heads, more or less thick, and then this somewhat restrains the degree of mobility and prevents, the naturalness of their behavior to be investigated. Particularly in non-human primates, this is where we know that the brain is closer to or more similar to the brain of humans, and it's worth investigating this higher level phenomenology related to behavior. This is a major limitation because primates are big animals, large brains. You cannot really attach a cable to their brain. If you attach any device on the body, they'll pull it out. It's much more difficult to do this kind of experiments. In addition, major limitation in any species has been the fact that one can conduct neural recordings, let's say wirelessly or in unrestrained animal, but you are limited. You don't really know what the animal is doing in real time, what the animal is seeing. So you don't have the sensory control that is accurate in real time. And for instance, we know that in hippocampus, we know that there's recent evidence that that visual input will shape the play cells. This is from Mayank Mehta's experiments in, I believe, rats, in which I found that there's tuning to visual stimuli even in hippocampus, right? Hippocampus is believed to have a place cell encoding. So if that is the case, then one could argue, for instance, that you should be able to, or you should control carefully the visual input, not only the location of the animal, right? So controlling essentially, or the more control you have on what the animal is seeing or sensing, the more accurate you can understand the neural representation and understand the relationship with behavior. So this has been a process that started in about 2012, and this is one, took years. We failed at the beginning, then succeeded, etc. This is an example. This is a project which was led by Russell Milton. He's now postdoctoral fellow. He's in Oregon after he graduated. So basically, Russell did an implant in dorsolateral prefrontal cortex, which is an executive cortical area using theta array. The advantage of this implant is that theta array can be chronically implanted. It's subdurally, you have artificial dura on top, you have a cable coming out, you have a transmitter. Will transmit this 400 channel wirelessly, the signal, right, to a computer, and you can. We had this overhead camera that is able to record the animal position in real time at any moment, not only position but also what the animal is doing. And in addition, we had the wireless eye tracker. In this project, the eye tracker was not as evolved as in later incarnations of this project, but nonetheless, it was a lower resolution eye tracker that, in principle, with such an eye tracker, is you're able to record. Basically, there are two cameras. There's a eye camera and a scene camera. So the eye, you always look at the eye, and then the scene always look at the world, and you're comparing the two views, and then you understand based on what the pupil is, how the pupil is moving, and what the scene is. You can know by calibrating this eye tracker, you can know what the animal is looking at. In this project, we didn't really take advantage of that, the visual input, but essentially, instead, what we did was in prefrontal cortex, we were able to track animal behavior. So we did this kind of longer term recordings for hours while the animal is in a larger cage, right? A plastic cage. And then we recorded the signals wirelessly in different states of wakefulness, such as quiet and active, defined by how much mobility the animal had. In the quiet wakefulness, the animal is barely moving. We have a threshold discrimination between successive frames, right? In this case, the difference between frame 1 and frame 2 is very close to zero. We have very few, say, white or gray pixels, whereas in this more active state, you find the difference between frames, successive frames, is much higher. So animal is actually performing something, is moving, is performing locomotion or raising the arm or being in a much more active state. In this case, the animal is just looking for food. So there's no specific task. So the goal of this project, which Russell has successfully accomplished, was to be able to predict the state of the animal by decoding population activity. So essentially, having a decoder that is blind to the state of the animal, you used to train this decoder using the data from the experiment and then test it blindly, and then we're able to distinguish whether the population could predict whether the state is active, quiet, or animal sleeping. And then this will allow us to look at which signals are most relevant for such decoding, such as the low frequency LFP signals or VG signals or spiking activity or other types of signals. We found the spiking activity can more accurately tell us the state of the animal. In fact, better than the LFP activity in this example. So anyway, our efforts to incorporate wireless technology into our experiments and to understand more complex forms of behavior has been, like I said, incremental, right? So this is the first incarnation of the project in which eye tracker was present. And by the way, attaching an eye tracker to an animal performing the task, in which case monkeys, this is an expensive piece of equipment. It's $30,000 just to buy this equipment, and really want to, make sure it's a good investment. So we don't want the animal to like bump its head or on purpose to pull it to destroy it, because then the animal does that on the cage. We just lost it in a second. So $30K investment. So we first trained using a dummy device. It took us weeks. When the animal is comfortable, we still continued with the dummy until it was super comfortable, and then we added the actual device because otherwise you just can't afford to do it. And we can, of course, our efforts now to increase the resolution of this eye tracker to really be to match the eye fixed, the head fixed type of recordings and of pupil and eye movements. All right, so just want to jump now directly to something I'm pretty excited. Paper published last week, in which you use this technology to investigate even more complex type of behavior. We have another paper that's coming out next month on foraging behavior, but this is social interactions. So in this case, we had two animals separated by transparent plexiglass piece in a larger cage that were essentially trying to perform a task. And the task was a social task in which they would simultaneously press buttons, and then a tray carrying reward will come to them. We call it cooperation task because they cooperate in order to get reward. And what motivated us to perform this experiment was the fact that, while social interactions have been investigated so far at the behavioral level, there is very little understanding of neural circuits underlying social behavior. Having said that, there is a wealth of information about single-cell recordings done in a variety of areas that are specifically social areas, related to motivation, to intention, to like so-called social neurons, right? Like mirror neurons that mirror the others, the conspecifics, activities, etc. However, in higher, let's say, higher carnivores and primates and humans, we typically engage in social interactions by first by using body language, right? We first make eye contact with the partner to understand whether there's something there's worth cooperating or not, right? So we make a lot of visual interactions before we even engage in social interactions, and there's a lot of evidence that this is the case in humans and primates. Now, exactly how this process unfolds in real time is unknown. And is unknown for obvious reasons. It's unknown because it has been impossible until now to be investigated in conjunction with neural recordings, because it requires high-resolution eye trackers that are synchronized with the recording of neural events, and this has to be done wirelessly in free environment exploration. In humans, it's difficult to do, or maybe it's not difficult to do the eye tracking part, but the invasive neural recordings are difficult to do for obvious reasons. In primates, the neural recordings are less difficult to do, or are possible to be done, but the tracking of the other things, such as the gaze of the animal in real time, and the behavior of the animal, and synchronize all of these events, has been impossible until now. So this is a project led by Melissa French, but there's a bunch of people here that help her accomplish this project. Essentially, what we did here was to have a dual UTA ray implant in visual cortex and prefrontal cortex using 100 electrode arrays, and then we have the transmitter sending signals from each of these from each neuron, recorded in both areas. So at the same time, the animal is wearing this eye tracker that is able to record, like I said before, the eye as well as the scene. And this is an example of different essentially points of fixation. We use deep lab cut to classify these different objects. The deep lab cut is based on machine learning that allows us in real time to, on the fly, to label different objects that are fixated by one of the animals. And by the way, in this recording, only one of the animals was implanted. The other animals was in its natural state. Now we have the ability to conduct dual recordings from both animals simultaneously and finally look at this type of interactions from both perspectives, essentially. So to be able to see if you can decode the other brain from one of the brains, essentially, and to understand the intention of the animal by decoding neural activity from this animal, for instance. Anyway, so let's see, I can show you this. This is a video. right? This will show you how the task unfolds in real time. So see here that one of the animals, this monkey is looking at this monkey, is already pressing the button, waiting for this animal to cooperate. The other animal makes eye contact with the other animal. There's a lot of exchange. And then finally, the other animal will press, both animals press simultaneously, and then the tray comes to them. They collect the reward. They get the reward, and the tray goes back, and then another trial starts. And by the way, in this experiment, there's no trial structure. It's a continuous flow of events. So we don't have the division between trials, and it's a good thing, I think, because we can do continuous time analysis of the neural and behavioral events. So we use, like I said, there's a sequence of events here that we carefully analyzed using a hidden Markov model in which we look at each event, so the succession of these events, which one comes first, second, etc. And then this allowed us to identify the important events in this task. What's most important? Looking at the other animal, looking at the rewards, etc. And then this would allow us to guide the neural recordings to target those specific activities to analyze actually what happens in social interactions. And the basic question here was the basic question was to have an understanding of how visual inputs or social decision or social or the encoding of social events that happens in visual cortex controls the activity or informs the activity of neurons in prefrontal cortex to guide the decision making. Okay, so how does the decision to cooperate is guided by what the animal is performing, essentially, what the animal is looking at, and how neurons are encoding that type of information. All right, so in this slide, what I'm showing here is that, first of all, there is a learning of cooperation during cooperation. So here we look at the cross-correlation of button pushes of the two animals. So cross-correlation captures the simultaneity of this the behavioral response. And as we see cross sessions from the first to last session, there's a sharpening of this correlation and also increase in the peak, indicating the fact that they learn to perform this interaction simultaneously. So when one animal presses, as animals learn the task, they tend to reduce the gap between the time of the push, which is also captured by this middle panel. This is mean push as a function of, this is the lag, right, in seconds. So the lag decreases in both pairs of animals, right, in a significant manner. And then the coordination of the pushes, temporal coordination, is also increased during learning. So this is to show that there is a learning, there's something going on here. The animals evolve from simply, making random fixations, looking at each other, random pushing of the two buttons, to actually doing this in a coordinated manner. And then, a difficult question, and this has been one of the, let's say, the dreams of neuroscientists, is to be able to record from visual neurons or neurons in other areas as the animal is seeing the world, right? So on the fly. And this will allow you to distinguish between so-called active and passive vision, right? Active vision meaning that, or could be any sense, active sensing, and more passive perception. So active sensing is when the sensing actually means something to you. So you're using it in some context, in which you're in an engaged task. You're doing something with that information, as opposed to just passively looking at stimuli. And I would say in sensory neuroscience, this research has been dominated by studies for decades in which animal is fixed on a chair, passively stimuli were presented on a screen, and people make careers using this type of configurations. But because technology was not really there to allow one to measure these events in real time, and as animal explores the environment, that type of knowledge was prohibited until now. So anyway, so this is another example of. So just to make a long story short, this is an example of both in areas V1 and V4 in prefrontal cortex. These are single cells encoding or responding to different types of stimuli, such as viewing reward, viewing a partner, or when the animal is pushing the button in different areas. So you see this anticipation of the button push in prefrontal cortex, but not in visual cortex, etc. So yeah, let me show you this movie. And I'll tell you why it is important. This is an example of what the animal is looking at and how neurons are responding. So we're using deep lab cut, we're labeling different parts of the world that the animal is inspecting before making a decision to cooperate. We're using this, like I said, to build this Markov model to understand the chaining of events, to understand what's important here, which one comes first, which one comes second, etc. And on average, visual events come before the button push as expected. But not all visual events are equally important. So essentially, this allowed us to identify or distinguish between social cues and non-social cues. So social cues are those visual stimuli that the animal is looking at, such as reward and partner, that are directly linked to decision making, right? To make a decision. Other, like higher probability, essentially what I mean with this, using this model of this social cues being looking at the transition probability between looking at this type of events to making a button push. And this allowed us to distinguish between social and non-social cues, such as viewing self-button or random objects like the floor, etc. And with that type of knowledge, we then went on to investigate how the population coding of this different type of events, social cues and non-social cues, evolves in time during time course of learning. What we found is that in both animals here, what we found in both areas, there's the learning effect here as well. So the accuracy with which stimuli are encoded increases in time during time course of learning. So in other words, the ability of the animal, the ability of the brain to distinguish between relevant and irrelevant stimuli is enhanced during time course of learning. We found that this ability is present for social cues, but not for non-social cues, in which there's a flat correlation, essentially, or there's pretty much no significant correlation between the accuracy of the decoder in either area, V4 or PFC, and the session number, right? Session number meaning whether there's effective learning or not. So if you see positive slope, like in this case, means that there is a learning effect. Or could be negative slope that is significant, which also indicates a learning, but in the opposite direction. But that's not what we observe. So the point here is that there is learning of, the ability of neurons in both areas to distinguish between different stimuli in the environment, which is different between social and non-social cues. Importantly, I believe this is the last slide I want to show, incorporates data, is that one main reason to record from at least two areas at the same time is to look at the interaction between areas, right? Network interactions. And there are different methodologies to do that computationally. In this case, we employed, now we're doing other analysis as well, but we employed this pairwise correlation method, looking at spike timing, the coordination between neurons in areas. So we did this both within an area, within V4 and within prefrontal cortex. And what we found is that the coordination of neurons, the rate of coordination increases as a function of, or as learning progresses in both V4 and the dorsolateral prefrontal cortex. We found this effect in both animals that were tested. Also importantly, between areas, this effect occurs. So we looked at the coordination between V4 and dorsolateral prefrontal cortex that is also increases during time course of learning. Importantly, it's not shown here, but it's shown in the paper. We found that specifically those neurons that are significantly temporally coordinated between areas in V4 and PFC have a higher decoding accuracy to decode relevant stimuli compared to the rest of the cells. So this tells you that this tells you something about the mechanism of this complex behavior, which is a social interaction, which is the fact that in time, neurons become more coordinated in time, and they transmit information most efficiently. And then you can ask, what is the type of information transmitted from visual to prefrontal cortical areas or executive areas? It is specifically the information about relevant social cues, because those are the neurons that are encoding stimuli more accurately compared to other neurons. And this is all documented in the paper, described quite well with a set of figures. For simplicity, I didn't show everything here. And also importantly, in the paper, also discussed the the strategy of the neural population to essentially perform this task. So initially found that only a subgroup of cells in each area is involved in decoding stimuli that are relevant for this task. But over the time course of learning, there is kind of redistribution of this type of information across pretty much all the neurons in the network. So there's more, I would say, egalitarian or communistic approach to information processing as learning progresses. So essentially, the bunch of few neurons that are initially more involved in the task will either through refinement of connectivity, local connectivity during time course of learning, but will essentially pass on this information to other cells. So the more cells are involved or recruited in this process. All right, so just to have a final slide about future challenges using these techniques. Now, wireless technology, we plan to have techniques to record from, now we're able to record from hundreds of neurons, but we plan to record from thousands of neurons, if possible, high temporal accuracy. There are many reasons why you want to go from hundreds to thousands. Over the past decade, there's been an effort to go from tens of neurons to hundreds of neurons, and there's no reason why you cannot go to thousands or even millions of neurons. For that, you need improved technology. Also, one of the challenges in the field is to be able to causally manipulate circuits during free exploration, right? So right now, we can have the animal in a kind of restraint paradigm to causally manipulate, so let's say optogenetically or electrically. But to do this on the fly as the animal is performing the task has been a challenge that hasn't been easy to perform. And I think there are very few attempts to do that, and there's a lot of improvement in technology to allow us to do that. Also, importantly, how multiple networks interact, not only two networks, but going to 7, 8, 10 networks over the entire brain, including thalamocortical interactions. This has been the dream and might come true in the next couple of decades through the improvement of technology. Importantly, can you translate or transport these technologies and approaches to the human brain, right? So to understand the human brain from a basic science standpoint, but also to use neuroprosthetic devices to improve function or restore brain function has been one of the main challenges in the field, I would say. Just want to thank my lab members. This is a relatively old picture a few years ago, but pretty much the lab right now is close to 15 people and trying to hire other people as well, the funding agencies, collaborators, and all of you for attention. And I guess I'm ready for more questions.